Good afternoon all. Hope you all are safe at home. I would like to introduce myself. I am Deepthi, Assistant Professor of Microbiology. First of all, I congratulate you all for clearing your first hurdle. Before going into today's class, I would like to highlight some current microbiological aspects. I am sure that all of you are keeping updated about COVID-19 pandemic that is reaping all over the world and our country right now. So I don't think there is any other better time than this to teach you guys about the importance of microbiology.
You listen about all the terms every day in media and newspapers. There are cases, there are positives, there are recoveries and even deaths. And also about viral particles, viral DNA and RT-PCR. You come to know about all these things through the media. And you will be wondering about the real thing behind all these terms. So in this subject, we are going to learn all these aspects. Right now, all over the world, the most busiest people in the medical field will be microbiologists. So this subject is so important in this era and we have to learn it. So coming to the importance of microbiology, without knowing microbiology, I can assure that you cannot even be half a doctor. Whether whatever field you choose, whichever specialty you choose. We all know infections are bound to happen or they're always there around us. We being Indians and we are living in a country where there is a hub of infections and infectious disease. So this subject is all about infectious organisms and mode of action, transmission and clinical scenario they are causing. So without knowing this, there is no medicine and I request your attention to the all subject as well as today's class.
To discuss about physiology or physical properties of bacteria.
Alright, we are discussing about physiology or physical properties of bacteria. So we are going through bacterial cell division, its growth and nutrition and its metabolism. First about bacterial cell division. We all know bacteria divide by binary fission. The process is described here. The nuclear body divides first and the entire division of bacterial cell by transverse septum or constrictive or pinching process. Next is about an important term, that is generation time or population doubling time. It is a time required by a bacterium to give rise to two daughter cells under optimum condition. Optimum condition means optimum temperature and optimum pH. It may vary. For coliform group of bacteria, the generation time is 20 minutes, whereas for tubercle bacilli it is 20 hours and for leprae bacilli it is 20 days. And the reproduction of bacteria is by a geometric progression. And if we plot bacterial population according to time, it increases and the population will decrease when the level of nutrients becomes low and the accumulation of toxin happens. Next is about batch culture and continuous culture. In batch culture, we are growing bacteria in a vessel of liquid medium. After few bacterial cell division, the growth may get arrested due to the accumulation of toxin and the depletion of nutrients. Whereas in continuous culture, we are using special devices to replenish the nutrient level and the removal of toxins. We are using chemostat and turbidostat for this purpose. Mainly we are doing this for industrial or research purposes. And another terms coming under this topic is total count and viable count. Total count is the total number of bacterial cells in a sample, whether it is living or not. The total count can be obtained by direct counting under microscope with counting chambers. Whereas viable count is the number of bacterial cells which are living or which are capable of multiplication. And it is obtained by pore plate method, surface viable count and Miles and Misra method. So if you are plotting the bacterial growth pattern in a graph, it shows 4 phases. First one is the lag phase, second one log phase, the third one is a stationary phase, and finally phase of decline. We are seeding bacterium in a medium. First is the lag phase. During this phase, the bacterial numbers will not increase. Only the size is increasing. And the lag phase is regarded as a time for the bacteria to adapt to a new environment. And during this phase, enzymes and metabolic intermediates required for multiplication are built up. And the duration of lag phase varies according to bacterial species, the size of inoculum, and the type of medium and environmental factors. That is about lag phase. Second is the log phase. Here bacterial multiplication occurs exponentially. And the third phase is a stationary phase. Here, as the name indicates, there will be no increase in bacterial numbers or bacterial size, mainly due to nutrient depletion and accumulation of toxic products. The number of bacterial cells formed equals the number of cells died. And the viable count is in equilibrium. And coming to the last phase, that is the phase of decline. Here the bacterial population decreases due to nutritional exhaustion, accumulation of toxin, and action of autolytic enzyme. This is the growth pattern of viable count, whereas total count shows only lag phase and log phase, no stationary phase, and phase of decline in the total count is seen. That is about bacterial growth curve. That is very important. Next is a number of cells, and its characteristics during these phases. In the lag phase, the size of bacterium will be maximum. During the log phase, the size become smaller, but the uniform staining characteristics is same. During stationary phase, the cells are gram variable, and irregular staining techniques they exhibit, and spores are produced, exotoxins and antibiotics are also formed. An involution forms are seen in the last phase, that is a phase of decline. Next is about bacterial growth and nutrition. For bacterial growth, a minimum requirement of water and sources of carbon, nitrogen, hydrogen, oxygen, and inorganic salts are needed. Next is about bacterial vitamins or growth factors. They are organic compounds essential for fastidious organisms. For example, thiamine, nicotinic acid, riboflavin, pyridoxine, folic acid, and vitamin B12. They are the bacterial vitamins needed for its growth. Other factors affecting the growth of bacteria are sources of oxygen, carbon dioxide, temperature, pH, light, osmotic effect, mechanical and sonic stress, and moisture and desiccation. And based on oxygen requirement, bacteria are divided into obligate aerobes, facultative anaerobes, microaerophilic bacteria, obligate anaerobes, and aerotolerant anaerobes. First is about obligate aerobes. They are strict aerobes which grow only in the presence of oxygen. For example, Pseudomonas aeruginosa. Next is about facultative anaerobes. They are originally aerobes but can grow even in the absence of oxygen. For example, E. coli. Third is about microaerophilic bacteria. They are bacteria which grow better in low oxygen concentration. For example, Campylobacter. Next is about obligate anaerobes, that is bacteria which are killed on exposure to oxygen. For example, Clostridium. The last one is aerotolerant anaerobes. bacteria which do not require oxygen for its growth and multiplication. Most of the medically important bacteria, that are pathogenic bacteria, coming under the group of facultative anaerobes. Next is about carbon dioxide requirement. Based on carbon dioxide requirement, they are classified as capnophilic bacteria, which need or which grow well in presence of carbon dioxide. For example, Streptococcus pneumoniae. Next is about temperature requirement. Based on temperature requirement, they are classified into three: psychrophilic bacteria, which grow best in low temperature, that is below 20 degrees Celsius; mesophilic bacteria, which grow best in a range of temperature between 25 to 40 degrees Celsius; and the last one is thermophilic bacteria, which need a higher temperature range, that is between 55 to 80 degrees Celsius. Next is about pH. Most bacteria grow well in an optimum pH of 7.2 to 7.6. Whereas lactobacilli grow well in acidic pH and Vibrio cholerae grow well in alkaline pH. About source of light. Most bacteria grow well in darkness, but certain bacteria, such as photochromogenic mycobacteria, can produce pigments on exposure to light. This pigment producing bacteria normally produce pigment on exposure to light. Next is about osmotic effect. This bacteria can withstand a wide range of osmotic variation. In hypertonic solution, it undergoes plasmolysis, and in hypotonic solution, it undergoes plasmoptosis. Other factors such as mechanical and sonic stress, moisture and desiccation can influence bacterial growth and multiplication. Next is about bacterial metabolism. Aerobic group of bacteria utilize glucose by oxidation, and anaerobic bacteria utilize glucose by fermentation. This process of oxidation that is described in Krebs cycle, you already know the oxidation and the fermentation process. These pathways you already studied in your biochemistry classes. And ATP production of bacteria usually occurs by substrate level phosphorylation.
Winding up today's session, hope you understood the content. Thank you for your attention.